Welcome to the Moss Report with your host, Dr. Ralph W. Moss.
Hi, this is Ralph Moss, and welcome to our podcast, The Moss Report. Our guest today is Robert Nagourney, MD of Long Beach, California. Dr. Nagourney is the founding director of the Nagourney Cancer Institute, and we want to jump right in and welcome him to the program. Hi, Dr. Nagourney.
Hi, Rob. Thank you for
Yeah,
having me.
no problem. Glad to be able to do so. So, for those who are not familiar with what you do and for what the Nagourney Cancer Institute has stood for almost, 20 years now, could you describe your unique method of testing drugs against cancer cells? Because it's really unlike anything that else pretty much being done, and certainly unlike the kind of testing that most of our listeners would be familiar with.
Yes, by all means. I'm a medical oncologist, and during my training and during my early years of practice, I questioned why we didn't have a better way to select active treatments. There was a great deal of interest at the time in genomics and genetics, and we had moved very aggressively into the study of the basis, the DNA basis of cancer biology, which has been very interesting and very exciting, but only gives us a veneer of information when it comes to the complex interactions of human biology. So we became interested in the idea that human tissue studies, that tissues disaggregated from cancer patients, broken down right out of surgical biopsies, could be exposed to drugs and targeted agents and metabolic inhibitors to look for injury and cell death. When we first started doing this, a lot of people were interested in growth based studies, how cancer grows and how to stop it from growing. And rather early on, we realized that growing cancer cells often introduced artifacts and changed the dynamic, and we became much more interested in shorter exposures with the endpoint of death, cell death. And it wasn't until many years after I'd started this work, particularly in leukemias, that we realized that cell death wasn't just a different way to get to the place you wanted to be, but was fundamentally different from growth inhibition. And that recognition, that realization, was confirmed by the description of apoptosis, and programmed cell death. So, quite by accident, we realized we had found ourselves in the middle of a burgeoning area of work, the study of programmed cell death, apoptosis, non-apoptotic cell death, and how that could select drugs for patients. So that's pretty much what we've been working on for the last couple of decades.
Yes. So you drew this distinction between necrosis and apoptosis. Some people say apoptosis, uh, programmed cell death. And I remember you once saying you could kill any cancer cell with bleach, but that wouldn't necessarily make bleach into a good cancer drug. Um, so there are more, let's say, more natural ways, right, to kill cancer cells.
Well, cancer cells, in a strange way, we think of them as being such nefarious opponents, but cancer cells are actually kind of stupid. I mean, what they do is they land upon some vehicle, some mechanism to stay alive, and they use it over and over and over again. And if you can find a very specific, select reason why a cancer cell is staying alive, like, for example, the luck that we've had finding things like epidermal growth factor mutations or ALK mutations. When you find a cancer cell that is so narrowly defines its survival advantage based on one particular gene, or the best example of all is chronic myelogenous leukemia. If you can find that one key to the cancer cell, you can eliminate the cancer. The thing is that cancer cells very often borrow lots of genes. It takes multiple genes for cancer to survive. They have a gene that allows for blood vessels. They have a gene that allows for evasion of immunity. They have a gene that allows them to get more nutrients. They have a gene that allows them to get more oxygen. So you're targeting lots of things, and we don't have the luxury of using a drug. So yeah, natural products, strangely, um, were were effective against cancer because they worked. I mean, it's just, betulinic acid from the birch bark or other substances work because they work, not because some smart guy at a pharmaceutical company came up with them, but because biologically they have an impact on the cancer biology. So yes, we have a kind of dictum that anything that works, we'll use any kind of drug or combination, repurposed drugs, anything that allows cancer cells to slide into their grave makes us happy.
And so your test is unique in a number of ways. One of the obvious ones, and it's sort of also in a way a limitation on it, is that it uses live tumor tissue directly from the person, from a biopsy taken of the person. Why do you insist on that when all these other tests, like genomic tests, so convenient? So it's so convenient that they can take readings off of slides or paraffin preserved samples of tissue, and you require an actual lump of tumor usually, or a blood sample in the case of certain types of cancer or some other bodily fluids, but you still need that cubic centimeter or so of actual living tissue. So why, if that's a disadvantage, why do you insist on that?
Well, the single most complicated technological advance in modern science and Western society and the world at large is a nuclear attack submarine. It's one of the most complicated things ever developed. And it's made up of multiple millions of components. Yet, the nuclear attack submarine, if you were to look at the diagrams of the nuclear attack submarine, it is very difficult to imagine that if you disassembled this nuclear attack submarine into its components the way we do with genes, we look at all the genes of a cell, it would be very difficult for someone, given all the genes of the cell the same way with all the components of a nuclear attack submarine, and assemble it into something that would go underwater and come back up. And the reason is because the complexity and the multiplicity of pathways in human cells are much more complicated than even the most complicated human technological advance. So the reason that we rely upon cells is because there is no analyte, two-dimensional gene profile, transcriptomic, phosphoproteomic analysis that even remotely approaches the complexity and the dynamism of living systems. So we are compelled to study human biology through the window of human biology because we just don't have the ability to apply big science to get these answers. Human tissue is too complicated. No one has the answer. And with the exception of a handful of genes that allow us for targets, very few cancer patients can be treated with just a gene profile.
I see. And you feel that that yields, right, by looking at the amount of programmed cell death that occurs over a period of some days in a test tube, basically, in a laboratory plate, that gives you a more accurate answer in terms of what's going to work inside the person.
Oh, yes. Oh, I don't think, I know. We have now done 10,000 patients and over the years have accumulated published peer-reviewed experience in a variety of diseases, including breast and ovary and lung and leukemias. And we compiled all that into a meta-analysis of over 2,500 peer-reviewed published experiences. And it turns out that if you use this method, if you apply cell death, you look what drugs cause cancer cells to die, you will more than double the response likelihood. And in fact, you also add statistically to survival. So we're very focused on using these approaches because, as I said, no analyte test, no gene profile, no protein profile can remotely approach that level of predictive validity.
Hmm. You've also said that cancer doesn't grow too much, it dies too little. Could you explain what you mean by that?
Well, yes, we can go back 150 years to the origins of our contemporary thinking about cancer. And what was the premise was that each cell comes from another cell. It was in Latin actually, but every cell gives rise to another cell, and that led to the concept that cancer was a abnormal cell growth. So for decades, centuries almost, we were studying how to stop cancer cells from growing. And that became the mechanism of studying cancer. Most people used cancer cells in tissue culture and cultivated them and propagated them and then looked to inhibit them. And then we developed drugs like, the alkylating agents like Cytoxan, and they damage the DNA, or the Taxol and zinc alkaloids, and they stop cells from undergoing mitosis, this ability to spring apart from one cell to two. Or we develop other classes of drugs, but they were all focused on one thing: cell cell DNA usage for growth, and cell growth. And I didn't read the paper today, but my recollection is that we haven't cured cancer, and we've been using these classes of drugs for 6 or 7 or more decades. So the question is, when you focus on something like that, and you say, well, if we've been doing this for that many years and it doesn't work, then is it possible that the premises and assumptions of our therapeutics are wrong? And I came to realize, using this model, this tissue culture model, that it seemed that if you could kill cancers, if you could stop them from growing, that those would be the drugs and things that work, that help the patient. And if you stop cells from growing, it seemed to be of no particular importance. And I had an experience with a laboratory that I worked with in the past, where we did grow cancer cells and measure DNA synthesis, and it had almost no predictive validity. So yes, it turns out that cancer doesn't grow too much; it dies too little. And if you want to cure a cancer, you have to come up with something that outright kills it, because cells that are not growing today can live to grow another day, but cells that are dead are dead.
Mhm. And you gave this very memorable TEDx talk, and you had many interesting things to say in that. It's available on YouTube. But one of the title of the talk was very intriguing. It was "The Future of Cancer Research Lies Behind Us," and I'm wondering if you could explain what you mean by that.
Yes, yes. Actually, I'm rather proud of that talk. I very much enjoyed giving it, and it reflected my underlying belief. I was a student of chemistry as an undergraduate, and I went on to, I don't want to brag, but I think, to excel in biochemistry. And I was convinced throughout my career that life was really fundamentally bioenergetic, that all of our, precise self-replication and all the stuff that we were focused on, and all the DNA analysis, really couldn't hold a candle to the understanding of fundamental bioenergetics and metabolism. And so I became increasingly interested in who had thought that way. And of course, you, as you go back through history, you find yourself realizing that the really great scientists of a century ago, now truly a century ago, were focused on how cancer differed from normal cells through metabolism. How did they utilize energy and metabolism to remain alive? Actually, if you think about it, cancer is using energy, nutrients, to remain alive, when the body as a whole doesn't want the cancer cell to stay alive. So the body is depriving the cancer of nutrients. It is depriving the cancer of oxygen. It is depriving the cancer cell of, of, um, of blood supply. And the cancer counters that by upregulating vascular endothelial growth factor, or HIF1 alpha. These are gene elements that allow the cancer cells to get more oxygen or to get more nutrients. And so, really, at the end of the day, the scientists who believed in the underlying metabolic basis of cancer were likely to be right, and that all of our discoveries and findings in cancer gene study lead us to the realization that most of the cancer genes are really metabolism genes. So yes, I think that the ultimate cure of cancer will be metabolic, and not cytotoxic or radiation, but more the inhibition of the cell's capacity to make and use energy.
Do you think that the origin of cancer is somehow a defect or an injury to the mitochondria in the cell? In other words, the portion of the cytoplasm, that the organelles inside the body of the cell, if you wish, that are responsible for generating energy. Do you see that as the locus for where this begins and where you have to direct your attack?
Well, the mitochondrion is the powerhouse of the cell, and it is fundamental to virtually all cellular existence, but life can um, can play out many different features. We know that some cancer cells borrow and utilize glycolytic pathways. We know, for example, that the serine pathway can generate some of the intermediates that the mitochondrion generates. We know that the pentose shunt, all of which occur extra mitochondrially, occurring in the cytoplasm. So, yes, I mean, I think that globally, cancer is a metabolic perturbation, and that the mitochondrion plays an extraordinarily important role in our metabolism. And so, yes, I mean, I don't think you could possibly address metabolism without addressing mitochondria, but it's global. I mean, it's both glycolytic and mitochondrial oxidative phosphorylation.
Hmm. And do you remember about 10 years ago that James Watson, who, of course, co-discovered the double helix structure of DNA with Crick, he published an editorial in the New York Times in which he basically renounced the genetic approach and endorsed a metabolic approach, and said that we have to sort of go back to the era of biochemistry, that we were on the right track with biochemistry, and that molecular genetics kind of shunted us off into a side area that you know had some relevance, but wasn't really the main show. He said that the molecular biologists have become the top dogs in the cancer field, and you can't really even get a grant to study just basic biochemistry of cancer. Would you agree with that?
Oh, absolutely. Um, I had a biochemistry colleague, a college student, very, very capable chemist. And he said, and I don't want to offend anyone, but he said that the molecular biologists were the quick and dirty artists of cancer science. And he felt that because we had looked behind the kimono and found the DNA that was aberrant in many tumors, that that was the way to go. And the problem is that in scientific research, people get a certain level of accomplishment. They make some breakthroughs, and then everybody rushes to that. And fairly soon, you get this self selection. So you start off with a few people doing genomic studies, and then they get some grants, and then they become full professors, and the full professors bring along with them junior professors, and the junior professors become the grant reviewers, and pretty soon you have selected only for one field of science. And I have said for decades as well that we have completely lost track of biochemistry. Ultimately, life is energy, and energy is biochemical, and in the human existence, it's metabolic. So yeah, completely agree. I think we went the mistake. I wrote a book, Outliving Cancer, some years ago, and I made the reference to the fact that we many years ago were on the verge of flying jets into space, and when Sputnik and Gagarin wound up in space, the entire American space program left behind our almost ready to fly into space jets and started slingshotting people up into space who would go up for an hour and land back in the ocean, or go around the Earth and land back in the ocean. And we did it because we had to get from point A to point B quick and dirty. But
Hmm.
the issue was that really what we needed was jets that could fly to and from. And we ultimately, and now again, are back in the process of coming up with jets that you fly up into space, because that was the right thing to do. So, yeah, I completely agree that it was a mistake to believe that we could peer into the gene. I mean, interestingly, when you speak of Watson, Francis Crick, who lived here in California, was very interested in understanding cognition by doing ultrastructural studies of brain tissue. So he had a kind of similar mindset that if you looked close enough at the structure of the brain, that you could understand cognition and emotion and things. And that didn't work, because these two-dimensional analytes are not the equivalent of the complexity of what they do when they work together. The neurons and the interactions of neurons cannot be captured by a picture of a neuron.
Yes.
We would like to take a moment to thank our Patreon subscribers for helping to make this program possible. If you would like to have access to exclusive content, including transcriptions, episode recaps, and special offers, become a patron today at patreon.com/mossreports.
I'm very intrigued by something you said earlier, which was that when you do your test, your ex vivo analysis or your functional test, well, you said that you can look at cytotoxic agents and you could look at targeted therapies and also look at sort of metabolic therapies. And I know you published an amazing case history of a woman who had, correct me if I'm wrong, but I think she had advanced lung cancer and it was advancing and metastatic. And she was treated or treated herself with metformin, or maybe you prescribed the metformin. I think you did. Um, and then she had a complete remission of her cancer. Not to suggest that that's going to happen uniformly in many cases or any cases, but it happened with her because the metformin, which is the most commonly prescribed anti-diabetes drug in the world, um, it seemingly was able to um, attack the energy potential of that tumor. So I wonder if you could describe that case a little bit and how does that work? I mean, my understanding of metformin is it's not a cytotoxic agent. It's an agent that would deprive the cancer cell of some of the glucose that presumably that it needs in order to perpetuate itself. How does your test work when you're testing so-called metabolic agents?
Well, again, the concept here is that all roads lead to Rome. That is, whatever injury or damage, if you can induce a state of instability in the cancer cell and it dies, you've won. So we're very open to almost anything that could cause cancer cells to die. And I have no dog in this fight. I don't really care what I test. The beauty of what I do is that I can test things in the test tube and I don't have to give them to people until there's a reason to do so. So in the case you described, which we published a couple of years ago, this was a 25 or 6-year-old woman from the most southern reach of Brazil. Her colleague in Sao Paulo, who was treating her for metastatic adrenocortical carcinoma, one of the most difficult cancers to treat. I mean, impervious to chemotherapeutics, very aggressive. She had been treated with a combination of etoposide, adriamycin, and platinum, had progressed. She had extensive disease. She had a large mass in her lung. And she called us and asked if we could help her. And I listened to the story and I listened to the history. And I said, no, I can't help you. And she said, well, I'm going to die of this disease. And I said, people with a disease like yours don't really respond to anything. I can't find anything for you. So she insisted on coming to California, and she flew from the southern reaches of Brazil all the way up to California with her family and her husband. And yes, we did an open surgical biopsy of the lung. And I must admit, I did everything I could to talk her out of this because I thought, we're not going to find anything. You have to have a surgery. You don't have American insurance. There's risk. Well, we happened to be working at the time with a pharmaceutical company from Japan who were working on mitochondrial inhibitors, and I was using as an internal standard fenformin, which is the parent compound to metformin.
Mm.
And we tested her, and she was brilliantly sensitive to it. I mean, shockingly. So I thought, well, that's amazing. So I called her doctor, a very lovely Dr. Peixoto in Sao Paulo, and I said, Dr. Peixoto, would you be willing to give this nice young woman metformin? Which she did, and the patient went into a durable remission. And it was so amazing that we then drilled down onto what the basis of that was. And I've never entirely figured this out, but working with a fellow named Bruce Ames, who was extremely and is a very famous biochemist, he and a Dr. Gatnaga, one from his group at Chori, which is the Children's Hospital of Oakland Research Institute, we drilled down onto this and felt that this patient carried a particular p53 mutation, which is a cancer survival gene, as it were, and that predisposed her to metabolic uh, targeting, and it worked so beautifully. She lived years with that treatment. Actually, it was just a stunning, astonishing experience, and we've had similar kinds of experiences with other quote unquote repurposed drugs. So again, I'm agnostic when it comes to what I use. I really don't care as long as there's some rationale and as long as we see a signal in the test, we'll recommend almost anything.
And what are you seeing? I mean, you have a unique platform from which to view, coming developments, because I'm sure you get asked all the time by patients to include this or that in their panel. What is it, 32 or 36 wells in the plate that you use?
Uh, Our standard well is 16, but we often do two or three of them, so
Oh,
we can do many, many drugs.
okay. So, do you see what looks promising to you at this point?
Well, I've really moved increasingly into the field of metabolism, and we published a paper a couple of years ago in breast cancer where we examined the metabolic basis of breast cancer. And we are just in the process of completing a manuscript. Tomorrow will be our final uh, author review. We have a meeting tomorrow with my colleagues at UC Irvine where I did the study, and we're going to get a paper soon into press in ovarian cancer.
Mhm.
And so we are increasingly of the opinion that cancer therapies will move toward depriving cells of energy molecules, of amino acids and of intermediates and of either proteogenic or non-proteogenic amino acids that are all part and parcel of metabolic machinery in the cell. So if if I were to say where would I love to be 5 or 10 years from now, I would love to be completely devoid of cytotoxic drugs and looking only at molecules that induce these metabolic injuries and cell death without poisoning the normal constituents of cells. You see, I started in our first moments of our conversation, I said that cancer cells are stupid. And they are, they're dumb. And they rely on processes that are actually targetable, like this woman with metformin. Her cancer cell was completely dependent on complex one of the mitochondrial generating adequate ATP. And when we blocked it, it died. So in this instance, if we can find those Achilles heels with simple molecules, you could imagine treatments that have no toxicity.
Hmm. Yes. Or you could even imagine treatments that repair the mitochondria, don't just kill them.
Yes, well, there's a big school of thought that cancer arises from mitochondrial perturbation, and that if you I'm sure you know this literature, where if you transfer cancer cell mitochondria into normal cells, they become malignant. So that all fits together with the idea that cancer is in one way or another a bioenergetic abnormality, and that the cancer may arise under conditions of metabolic stress. In fact, that's exactly what our breast cancer paper and what our current ovarian cancer paper has shown is that we are all carrying certain metabolic predispositions. And so the question becomes really interestingly, something you'd be interested in, is could you identify the people who are manifesting those perturbations, those sort of predispositions, and reorient their well-being before they get the cancer? I mean, that's a unbelievably exciting area that we're trying to just break into is not just treating cancer patients, but preventing them from getting cancer by identifying what it is that's leading to their cancers. And again, that's extremely early, but at least theoretically possible.
Yes. And I wonder if you could explain what you mean when you say that that can when you and we talked about this the last time we were together at the ASCO meeting, but when you disaggregate or break up the tumor sample that you get, and then you break it up into individual cells, most people would think that those cells would just sort of sit there, be sort of waiting for you to perform the test. But something very, very unusual and almost mystical happens. And maybe you could explain that about the microspheres. I find that so fascinating.
Well, the interesting thing, and I was guilty of this because my first work, my original 300 plus studies were conducted in hematologic tumors. And as you know, bloodborne tumors are like abnormal cells bouncing around in the bloodstream.
Yeah.
So they become sort of autonomous tumors. leukemias and lymphomas are circulating. CLL cells and acute lymphoblastic leukemia and acute myelogenous leukemia cells are kind of entities unto themselves. So when we first started doing this work, we would isolate the cancer cells from the blood. It was easy, reproducible. We could study all kinds of interesting molecular biology, and we could run all kinds of stuff. So we were doing flow cytometry, and we were looking bioenergetics, and we were running all stuff. So it was really exciting and really good. And using that, we were able to apply the technology, and we were able to identify what proved to be the curative therapy for hairy cell leukemia called chlorodeoxyadenosine. And we came up with some great combinations like alkylating agents with fludarabine that proved to be really effective in CLL. And it worked really, really great. And I was very pleased with my progress. And then I decided, well, since most people die of breast and colon and lung cancers, that I wanted to test those patients because it was 90% of the patients we were seeing had these solid tumors. And so we migrated into solid tumors, and I applied the rather simplistic idea that I would disaggregate the cancer cells from the lung patients and the colon patients into similar single cell suspensions. So we would aggressively chop up our tumors, and we would use enzymatic digestion, and we would use these density techniques to isolate single cells. And when we did that, the drug responses and the drug profiles stopped making sense. Like the patients didn't respond, and we didn't get good data, and we didn't get good outcomes. And so my techs began to work with me to leave the cells in larger and larger aggregates. So instead of doing these density centrifugations and isolating single cells, we began to do clumps or clusters. And those aggregates of cells gave us a little better window. And then we began to use larger clusters until we got to a kind of optimal size, somewhere in the range of about 50 to 70 more or less cells, seemed to recreate the biology of the tumor in the body. That is, it looked like the human body, but it was still penetrable by the drugs of interest. So we could get, we could, pharmacokinetically and pharmacodynamically, we could create the state of drug exposure in these aggregates while the aggregates still thought they were in the body. And
Hmm.
that became the model. That's these human organoids or microaggregates that we have studied over the last well
Yeah,
over 10 years.
right.
And those have turned out to be extremely predictive of outcome. And so yeah, we really landed in on what I call the ecology of cancer biology. And it means that you need the blood vessels, and you need the cells to talk to each other, and you need the cells to talk to the proteinaceous material called the stroma, and you need the immune cells like the macrophages and the B and T cells, and you need the cytokines. So it isn't a seed, it is a plant growing in a soil.
It's a
And
sprout.
that's really our model.
It's the sprout of the cancer in a way. It's a
Right,
very pure,
right.
and it's so amazing to look at your website, um, which is, I think, NagourneyCancerInstitute.com. Would
Uh-huh.
that be right? And
Yeah,
you
yeah.
have some pictures there of some different tumor types, and actually, these individual cells start to reorganize themselves into little tumors characteristic of the organ that they came from,
Yeah,
which
yeah.
is
In
phenomenal.
in fact, I did some work with a pharmaceutical company developing a proteasome inhibitor many years ago. And so I was traveling somewhat regularly to Cambridge, Massachusetts, and I used to really enjoy the lectures I gave there because they had a collection in the company who were pathologists by training, and they were part of the developmental program doing these small molecule developments. And I really liked when I would go and lecture because I would show pictures of my microaggregates, and I would turn to the back row in the auditorium and say, "Okay, what's this tumor?" And the guys could look at these little microspheroids and say, "Oh, that's renal," or "Oh, yeah, that's colon." I mean, you could actually these little clusters
Yeah,
look exactly like the tumor of origin.
yeah. So this is another level of sophistication, if you will, of being then directing drugs or metabolic agents because you're actually going up against that person's tumor. That's sort of the essence of the person's tumor, not just a isolated cell. So I think that's so fascinating. Well, our guest today is Robert Nagourney, MD from the Nagourney Cancer Institute in Long Beach. Has a wonderful website. Everybody listening might want to do what I do, which is to sign up for his blog, because once a month or so, Dr. Nagourney writes a fascinating blog, usually centered around an extraordinary case, a remarkable case of somebody who responded to treatment using his unique method of testing drugs and metabolic agents and targeted agents outside the body. So you don't have to be the guinea pig, as it were, uh, to find out whether or not this drug is going to work or is toxic. So, as always, Dr. Nagourney, great pleasure to talk to you. And for Moss Report, this is Ralph Moss.
Thank you very much, Ralph.
Thank you for listening to The Moss Report. Visit our website at themossreport.com and subscribe to hear more discussion of cancer treatment options, alternative research, and more. For more information about Dr. Moss and his work, including scheduling phone consultations, go to mossreports.com.